glycogen, coordinated regulation of glycogen synthesis and degradation. Glycogen synthesis is called as glycogenesis pathway. It is the process of synthesis of glycogen. If the glycogenesis has to be defined, it is the process of synthesis of glycogen for storage. It takes place in all animal tissues, but is especially predominant in the liver and skeletal muscle. So glycogen storage, in the last topic, we have seen that it is stored mostly in the liver and skeletal muscle. So glucose is converted into glucose 6-phosphate by glucokinase. So here in the liver, instead of hexokinase, we have the glucokinase. So that will convert the glucose to glucose 6-phosphate using the enzyme glucokinase, and then it is converted to glucose 1-phosphate by phosphoglucomutase, which then convert the glucose. It is transferring to the nucleotide sequence, so that now we get the sugar nucleotide, UDP glucose. So that is the immediate donor of the glucose by the action of UDP glucose pyrophosphorylase. So that is shown in the image. You have the nucleotide, triphosphate, then sugar phosphate is there. The product is the sugar nucleotide diphosphate sugar. So one phosphate is removed from here and one phosphate is removed from the sugar phosphate. So those two phosphate will form pyrophosphate. Then finally, this UDP glucose transfer the glucose to the existing glycogen primer. We'll be having the glycogen, stored glycogen already. Now the newly added glucose via the UDP glucose will it will straighten the chain. It will increase the chain length, and even wherever it is possible, it will form the branches. So where it is attached, the glucose which is transferred from UDP glucose to the existing glycogen happens at the non-reducing end. So in the diagram, you can show the non-reducing end in the left side. So there only the new glucose is added from the UDP glucose. This is the process called glycogenesis. The other process, glycogenolysis, is just opposite. From the glucose, it is converted into glycogen. And glycogenolysis is the breakdown of the glycogen at need. It is done by glycogen phosphorylase enzyme. The glycogen in the muscle provides a quick source of energy for either aerobic or anaerobic metabolism. It can be exhausted within an hour during the vigorous activity. So liver glycogen serves as a reservoir for glucose for other tissues when dietary glucose is not available. And even during or between the meals, during the fasting, it is important that the brain should get the glucose continuously, which cannot use the fatty acid as fuel. So liver glycogen is depleted in 12 to 24 hours. In human, glycogen stored is far less than fat, but fat cannot be converted into glucose. It cannot be catabolized anaerobically. So these are the drawbacks in using the fat for energy molecule. So glycogen is the immediate source of energy, immediate source of glucose. It will give the glucose and that can undergo the glycolysis. And through this, the energy can be utilized by all the tissues, including the brain. Glycogen, as you know, that it is having a linear chain as well as branched chain. So linear chain is connected by alpha 1 to 4 linkage, and branched chain is linked by alpha 1 to 6 linkage. So now if you want to utilize this glycogen, the glucose need to be removed one by one. At the same time, at the branch point, this branch linkage alpha 1 to 4 also should be broken down. So we have two sets of enzyme. One set is just debranching this linkage, debranching enzyme. Then after debranching, one by one, the glucose will be removed from the glycogen in the form of glucose 1-phosphate by the respective enzyme. And then the glycogen debranching enzyme cleaves the branch point to release the glucose 1-phosphate. Phosphoglucomutase convert the glucose 1-phosphate to glucose 6-phosphate. So it is just changing the position of phosphate from first carbon to sixth carbon. So always this type of shifting the position is done by mutase enzyme. So here also phosphoglucomutase is helping for the conversion of glucose 1-phosphate to glucose 6-phosphate. So in that way, the glycogen is now eliminated as glucose 1-phosphate. This can be converted into glucose 6-phosphate, and this can enter into the glycolysis pathway. So now we have seen the glycogenesis one side and glycogenolysis another mechanism. Both are just opposite flux. And this opposite flux of the carbohydrate metabolism or glycogen metabolism is coordinately regulated. Just like glycolysis and gluconeogenesis, this is also regulated by set of enzymes and by allosteric regulation and by hormones. In addition, the metabolite, cyclic adenosine monophosphates, cyclic AMP also will regulate. So let us see which are the enzymes regulated. The first enzyme regulated is the glycogen synthase, main enzyme, glycogen synthase, which will help for the synthesis of the glycogen or glycogenesis. So it is stimulated by glucose 6-phosphate. If you have enough glucose 6-phosphate, glycogen synthesis happens in addition to glycolysis. So this glucose 6-phosphate will increase when blood glucose level is high. So when blood glucose level is high, you get more glucose 6-phosphate, and that will induce or stimulate the glycogen synthase enzyme. So that will cause the glycogen synthesis. Next main enzyme which is under regulation is phosphatase 1. So you can see the glucose 6-phosphatase. Phosphatase 1 is activating the glycogen synthase by dephosphorylating it. So here mostly when certain enzymes, most of the enzymes will be activated when a phosphate is added. But here if you look at when a phosphate is added to the glycogen synthase enzyme, that is inactivated. And then phosphorylase kinase, that also will activate the glycogenolysis, and it is enhanced by the calcium level or by phosphorylation. Either by increase in calcium level or by phosphorylation, it can be activated. And then hormones, as usual, just like in the glycolysis and gluconeogenesis, insulin activate the glycogen synthesis, and then it increase the glycogen synthase enzyme in the liver and muscle, whereas glucagon in the liver only, it activate the glycogen phosphorylase, which break down the glycogen to glucose. And epinephrine, it is activating the glycogen breakdown. Glucagon, epinephrine, both are having similar effect only. But what is the difference, that I will show you in the next slide. And in addition to these hormones and these enzymatic regulation, the cyclic AMP, you can see in the diagram, cyclic AMP is inhibiting, that is minus symbol, inhibiting the glycogen synthase, and it is activating glycogen phosphorylase, so that the glycogenolysis is activated and glycogenesis is inhibited. Similarly, the insulin will suppress the level of cyclic AMP, whereas glucagon and epinephrine both will enhance the level of cyclic AMP. If cyclic AMP is increased, automatically the glycogenolysis happens and glycogenesis will be inhibited. This is the main coordinated regulation happening between the glycogen synthesis and glycogen breakdown. So the enzymes are under regulation, mainly the phosphorylase kinase. It will convert the phosphorylase B, which is less active, to the phosphorylase A, which is active. Here the phosphorylation is giving activeness to the enzyme. That is done by phosphorylase B kinase, and this is enhanced or this is stimulated by hormones glucagon in the liver and epinephrine in the muscle. That is the difference between the action of glucagon and epinephrine. Both are having similar function, similar role, which is just opposite to insulin role. But glucagon mainly in the liver and epinephrine in the muscle. Glucagon will act in the muscle as well. And what is happening here, you can see in the next illustration, next image, insulin is inhibiting the glycogen synthase kinase 3, and so that the glycogen synthase activation will be suppressed. And it is the glycogen synthase A is activated by insulin and then glucose 6 phosphate and glucose, whereas it is inhibited by glucagon and epinephrine. This glucagon and epinephrine is antagonist to insulin. So it will stop the glycogen synthesis and promote the glycogenolysis. Here you can see the cascade of hormone action. The main hormones which regulate the blood glucose are epinephrine and glucagon. Insulin role, you know that it always suppress the level of blood glucose, whereas the epinephrine and glucagon at stressful condition, at fasting, at exercise, it has to maintain the blood glucose level. For that, it will enhance the glycogenolysis. How it happens? In the muscle, epinephrine will bind to its receptor. The right side image you can see, it will bind to its receptor. And glucagon, it will bind to its receptor in the hepatocyte, that is liver cells. So both will bind to its receptor. It will activate a G protein. It is a G protein coupled receptor, and it will convert the ATP to cyclic AMP. So now cyclic AMP level is high. Always when cyclic AMP is high, it will promote the glycogenolysis. It will suppress the glycogen synthesis. So this cyclic AMP, how it does? It will activate another enzyme, inactive protein kinase A to active protein kinase A. So the signal is amplified now from 20Hz. Here this activation happens in 10Hz. So you multiply it. So you will get the enhanced signal. And now this active pKa will convert the inactive phosphorylase B kinase to active phosphorylase B kinase. Now again signal is enhanced to 100Hz times. For one hormone only this signal amplification happens. That you should understand. For one hormone, the signal amplification happens. And this is activating the glycogen phosphorylase A. Now it is 1000Hz. And this phosphorylase A is converting glycogen to glucose 1-phosphate. So 10,000Hz molecules are produced. So from one glycogen, 10,000Hz glucose 1-phosphate is produced. All this happens just when one epinephrine bind to its receptor or one glucagon bind to its receptor. So in that way, this glucose 1-phosphate then converted into glucose going for the glycolysis. It is going to the blood system now. From the muscle cells, from the hepatocyte, now this glucose is coming to the blood so that the blood glucose, normal blood glucose level is maintained. It is also undergoing the glycolysis in the muscle. Muscle, we need the energy. So the same glucose 1-phosphate converted into glucose and or glucose 1,6-phosphate enter into the glycolysis, and it is helping for the muscle contraction. So all this happens when we don't have much glucose in the body from the diet or from the glycogen. This happens. So this is regulated by the hormones, glucagon, epinephrine both in the liver and epinephrine in the muscle. And this little difference is there in their action, not only their receptor and then activation step. Here in the liver, if you yes,
Ma'am, next class has been started, ma'am, now. Excuse me,
okay,
ma'am.
then I'll stop. Okay,
Okay.
okay.